My name is Shabnam Suknyandan. This lesson is brought to you by the Gauteng Department of Education in collaboration with the Sibona Discovery Center. So today, learners, what we're going to look at is actually the difficult questions that appear in paper 1. Remember, the majority of the topics are actually in paper 1, and if you can master the technique of answering the questions with regards to the topics, you can really do well and you can pass life science with a distinction. So we're going to have a look at some of the difficult questions in paper 1. And we're going to now look at the multiple choice questions, right? And you know that the multiple choice questions always have the answer. So you can actually score full marks if you know the actual skill of answering the multiple choice. So let us look at it. The technique in scoring total marks in MCQs is you must have studied the content of a topic very well. In other words, if you know the content of the topic, remember what are the topics in paper 1? Yes, it is the human eye, the human ear, the brain, the spinal cord, right? That's the nervous system, or we call it responding to the environment. Then you have your endocrine glands and their hormones and how they actually are kept, they release substances to keep it constant, which is homeostasis, right? And then you have your human reproduction, which is your male reproductive system, female reproductive system, right? The gametes, and then the reproductive strategies. So that all forms part of paper 1, right? So now we're going to think, if you know the content, you can do well. Then you must identify the key words in the stem of the sentence. So what are the key words that lead to the alternatives in the multiple choice? Right. Then also you must be able to eliminate the incorrect answer. Somewhere the incorrect options will be given and you need to know, I must cross this out because definitely it's out. And now I'm left with the option of these two. Between the two that left, look at what is the closest one to the main idea in the multiple choice question. And you find that, let's look at the multiple choice that was, first of all, very poorly answered in the prelim paper due to having many criteria, and then you have to look at the criteria and then choose from the alternatives. This one here now related to the eye, and many learners, although it looked easy, they found it difficult because of it was a lot of criteria involved. See, the pupillary reflex. So the minute you see pupillary, already you know this question deals with the eye. It deals with the pupil. Reflex means how it's going to respond. Né? And is the ability of a typical human eye to adjust to different light. Remember when we talk of the pupil, it's an opening, and with the opening, it allows light to pass through. Different light intensities in the environment. Which combination of these processes will occur in a typical human eye when the light intensity changes from dark to bright light? So where you're going from is from a dark room to a bright room. So don't be confused. They want to know what happens when you're going into a bright light. The change, you're in a dark, now you're going to bright light. So what are the changes? Now let us see, there's various options here. So you must tick off on your question paper. Which one relates to bright light? Bright light. So radial muscles contract? No. Remember, in bright light, it's going to be CC. Circular muscles contract. So you know that. Now you look for it. Oh, here's circular muscles contract. No, that one there is for the dark, radial muscles contract. Pupil constricts. Yes, in bright light, the pupil becomes smaller, or we say constrict. So you tick it off on your question paper. And then RR, the radial muscles will relax. Yes. Circular muscles relax. No, that is for dark light. So that one's out. And obviously, your pupil will dilate, got to do with the dark, because during the day, your pupil constricts. So the options are 2, 3, and 4. So you look, where do I have 2, 3, and 4? I have 2, 3, and 4, and the answer is A. And that's how you are able to analyze your questions and do your multiple choice well. So you see, immediately you can eliminate, and from whatever's left, you look for the options and then you get the answer. So it's not difficult at all. There the answer is A. A only. Okay, but in multiple choice, you don't say only, it's just A. Now we're going to look at another question that looked simple but was very poorly answered because the children interpreted it incorrectly. The diagram below shows stages of development. Immediately you know what is this. This question is coming from what? Reproductive strategies. Right. Now here you can see in diagram A, there's a little chicken with its eyes closed, no feathers. It's unable to move. Can you see? It's completely helpless. And here's a baby crocodile which has just hatched out of its egg. You can see its eyes are closed, but its mouth is open. But the fact that it can come out of the egg and walk on its own, this one is quite prepared because it actually tore the shell and walked on its own. But this one here, the eyes are completely not even be able to walk, no feathers to fly, so it's completely stationary. So the type of development where a small amount of yolk was available to the embryo. Remember the question previous to this is which diagram, A or B, shows you altricial development. Now what is altricial? When the eyes are closed, then they said no. Even the crocodile's eyes were closed. No feathers. Crocodiles don't have feathers, but you can see they have a skin. This relates to A. There's no feathers. Eyes are closed. Unable to move. Completely dependent on the parent for its food. So that is A. That was altricial. Now when they ask the type of development where a small amount of yolk was available to the embryo, because learners said altricial is A, then they thought no, we can't say for the small amount of yolk, it can't be A again. It has to be B. It was wrong. The answer is still A, because these little chickens, right, cannot move on its own. Its eyes are closed. There's no feathers at all. That means this one is altricial and this one had little yolk. So even for this one, the answer is going to be what? The answer is going to be A. Okay? So it doesn't mean if you took A in the first part of this question that B should be the second part. No, it can still be the same diagram. Okay? So remember that B, this crocodile is showing precocial development. Precocial means it's prepared, it's able to walk. It can even go and look for its own food. So this crocodile is showing precocial development, and the fact that it can walk out of the egg means it's able to move, and that's why it was prepared. And if it was prepared, it came from a lot of yolk. Okay, so that's B is precocial development, A is altricial development. Now comes another question which looks very simple, but almost every learner in every school, when we ask them, which question did you have a challenge with? 1.5.3. They said, name the tissue of the uterus that contracts to enable the woman to push the baby out during birth. So many learners thought it's part. I have to look for part in the system. But you know, there's a uterus wall, there's a fetus, the endometrium, the bladder, the pubic bone, the colon, there's a vagina, there's a cervix. So which tissue can push the baby out? And although you knew that cervix is which dilates, the uterus contracts and the cervix dilates to allow the baby to move out. Many learners wrote cervix, but cervix is not a tissue. It is a part, but it's not a tissue. So the only tissue, learners, that is able to contract and relax to bring about movement, to push the baby out, is going to be what tissue? Muscle. So the actual thing here was, what tissue is going to cause the baby to be pushed out? Muscle tissue. You could also say myometrium. Okay, but we don't normally learn that. But muscle tissue is the muscle that contracts. Remember that. The minute you use the word contract, you must know the tissue that got to do with that will be muscle. Because only a muscle contracts and relaxes, like the radial and circular muscles of the eye. They contract and they relax and they bring about the opening or the movement of the pupil, to dilate more or to become smaller. Right. So then here is that question was poorly answered. So please understand that tissue means muscle tissue. And now we're going to look at other diagram, which is very common in paper one, and that got to do with your normal menstrual cycle. Always when you get a picture, remember whatever you look at, your brain must now manifest. What is this? Which actual topic is it coming from? Okay, this is the female reproductive system. Now that it's registered in your mind that this is the female reproductive, got to do menstrual cycle, it must do 28 days, and then you analyze the diagram. Always read your diagram. They give you clues to the answer. Remember, because you're under stress, you may just look at the picture and you may just write. But sometimes what they may make some small changes which you're unaware of because you didn't read, and then you find that you will write an answer that is incorrect, and yet the answer is blatantly can be observed on the question paper. So let's look at it here. The first half shows you pituitary gland hypophysis. Another name for pituitary is hypophysis, and the hormone levels. So this 1 and 2 only got to do with hormones released by the pituitary gland. And what is it? It can only be FSH or LH. Now analyze it. Before you look at your questions, analyze the diagram. So the questions become easy. Oh, that's a pituitary hormone. Okay, that's number one. Number one is a solid line. And then number two is a dotted line. You're analyzing it. Oh, number two goes very, very high. It peaks on the 14th day. And from your knowledge, from the content you learned, you know on the 14th day, which hormone peaks from the pituitary? It's going to be LH. So automatically you know that the one that peaks is very high is number two, and that is going to be LH. So obviously the only other hormone released by the pituitary involved in the menstrual cycle is going to be what? FSH. So you put FSH, and what will that be? Number one. Let's see if it's correct. As we see follicles are growing, can you see something is causing the development of the follicles? Who is that? That is FSH. It can come here, and then just before the 14th day, the most mature follicle formed, which is the Graafian follicle. So it formed on the 13th day. That's why you can see there is a sudden sharp increase in FSH on the 13th day, because your mature Graafian follicle formed. And who caused it to develop? FSH. And then on the 14th day, we know that that Graafian follicle bursts. Can you see? On the 14th day, that's when ovulation occurs. So the Graafian follicle ruptures. And then what's formed? Your corpus luteum. And you know that in the second row here, this is how your follicles are developing. Primary follicle, secondary follicle, Graafian follicle. Then ovulation. Then what are these? Your corpus luteum, which is your empty follicles. And that is showing you the development of the follicles. And then here now in the third row, you can see they are talking about ovarian hormones. That means hormones from the ovary. It cannot be LH, it cannot be FSH. So what are the two other hormones you learned about in the menstrual cycle? It is estrogen and progesterone. Why? Because these two hormones are released by the ovary. But what releases estrogen? E for ovary, estrogen, O for estrogen, there's the O that's silent. And you must remember that it is the follicles, which also has an O, follicles in the ovary that releases estrogen, right? And you can see now, as the follicles are developing, what is happening? Number 4 is also beginning to increase. So number 4 is going to be your estrogen. And then automatically, after the 14th day, you're going to analyze that my corpus luteum formed. And once the corpus luteum forms, what does it secrete? Progesterone. So what increased or peaked after the 14th day was number 5. So number 5 is going to be progesterone. So here is your 2 ovarian hormones. You already know now. On your script, you write there. That's for progesterone. and that one is my estrogen. And that 0 to 7, what is showing here? You can see now the endometrium is busy breaking down. That here is menstruation. And then from the 7th to the 14th, the endometrium is building up. Who is building it up from the 7th to the 14th day? There's no progesterone. The endometrium is becoming thicker, but by who? By what hormone? It's only by estrogen. So 7 to the 14th day, as the follicles are growing, they release estrogen, which thickens the endometrium. But from the 14th to the 28th day, which hormone is responsible for the thickening of the endometrium? Now it can't be any follicles now because the follicles are empty. So the empty follicle starts developing the endometrium even further. And what is that empty follicle? Corpus luteum. What hormone? Progesterone. So from the 14th to the 28th day, it is progesterone that thickens the endometrium. Just till the 28th day, not for the whole month of all the months of pregnancy. The corpus luteum is just too tiny to secrete progesterone for 9 months. Remember, what takes over progesterone function after that is going to be the placenta. Okay? So you find here now, now you're going to look at your questions, right? Give the name and number of the hormone that stimulates the development of the follicle. It's going to be FSH. And then maintains the endometrium between day 7 to 14. It's going to be estrogen. Okay? So development of follicles is FSH and endometrium building up 7 to 14 is estrogen. Again, the same graph, right, on the menstrual cycle, very common in your books, in the Mind the Gap, in your DBE books, you find the very same menstrual cycle diagram, right? So study the diagrams and graphs below, which shows the hormonal regulation of the female reproductive cycle. So in other words, these 4 hormones are controlling the entire menstrual cycle. Now let's look at the question. Explain the function of the corpus luteum. Remember, you've already analyzed your diagram. Here's the corpus luteum. What is its function? You said to secrete progesterone to maintain pregnancy. Secretes progesterone to maintain pregnancy or to maintain the endometrium during pregnancy. But it is to maintain the endometrium during pregnancy. That is the function of progesterone. But remember that for the entire pregnancy, for 7 to 8, let's say from 3rd month to the 9th month, it's the placenta that produces progesterone. But till the 28th day, it's the corpus luteum that produces progesterone. But progesterone function is what? To maintain the endometrium. Now, describe how the corpus luteum is formed. Very poorly answered in the prelim paper. The learners had to talk. They cannot just say that the Graafian follicle bursts and forms the corpus luteum. Many wrote that and it was wrong. Okay? So they had to say what actually from the beginning, that it is the pituitary gland which secretes LH. The pituitary secretes LH. And then that stimulates what? The Graafian follicle to rupture or burst. It stimulates the conversion of the ruptured Graafian follicle, or the Graafian follicle to burst and rupture, which then develops into the corpus luteum. Okay, learners, so this always comes in the exam. What is it? That it is the pituitary gland that secretes LH. The LH will move into the bloodstream. I'm giving you even better answers now. That the pituitary gland secretes LH, which is a hormone which enters the bloodstream to go to the target organ called the Graafian follicle. The Graafian follicle will burst and release the ovum called ovulation. Put everything in your answer, and then the ruptured Graafian follicle will be called the corpus luteum, which secretes progesterone. Right. So that is the full answer of how the corpus luteum is formed. And then you go to the next slide, right? Now we go to the male reproductive system. And this is a very, you can either get the front view of the male reproductive system or you can get the side view, right? One of the two views, or sometimes they can even give you both the views in the paper and ask you questions on it. So now, children, whenever you get a diagram, always, very, very quickly, during your reading time, make a mental note of what are the labels. Oh, A is my vas deferens. B is what? B is going to be my bladder. Né? And then here's my C, that one that looks like a balloon, like a leaf. That's my seminal vesicle. The one below my bladder, B for bladder, there, and below my bladder is going to be what? My prostate gland. And then here's the urethra, which comes into the penis, and it's a common passage for what? Urine and semen. You don't say sperm. Sperms don't just move like that out of the urethra. They are always found in a fluid. The fluid plus the sperm cells is called semen. So that's the urethra. Then here's the testes, which lies outside the male body. And then it's protected by a little sac-like structure there, which is G. And what is G? It is the scrotum. And what is F? It is the testes. And what do you know? That the testes is stimulated by the hormone testosterone. That's the male secondary sexual hormone that is responsible for male secondary sexual characteristics like formation of the beard, development of spermatozoa, enlarging of the penis, pubic hair, chest hair, development of mustache. All those characteristics are brought about by testosterone. And then the testes is the one that secretes the testosterone. And what kind of cell division occurs in the testes? It's meiosis. Now you've analyzed everything about this diagram. Now that you know everything, you look at the question. Explain the function of G. And look at the word explain. It is highlighted in red. The reason is whenever you have a question saying explain, you need to use a cause and effect answer. Cause and effect means what is it? Give a statement and give a reason for your statement. That's the same as saying cause and effect. Okay, explain the function of G. Now G is the scrotum, and always, learners, write. When they ask you for a letter, write down. Show that marker that I know what G is. You say G is the scrotum. Whether that's in the memo or not, but you show them that you know what it is. G is the scrotum. So what's the function? It protects the male testes. You can't just stop there. It protects it. So how did you explain that? G is the scrotum. It protects the male testes because it lies outside the body. You see, it's not inside where it can be protected. Because it's lying outside, you need something to protect the testes, and that is the scrotum. So explain the function of G. Protection of the testes because they are held outside the body. Next question, or you could say the scrotum protects the testes, or it holds the testes outside the body to ensure that they are at a lower temperature for optimum healthy sperm production. So you can say that the scrotum protects the testes because the testes lies outside the male body, because the testes needs to be kept at a temperature 2 to 3 degrees, 2 to 3 degrees lower than the body temperature. Remember, if the body temperature is 37.5 and if that testes is going to 40 degrees, if a male has a fever, all his male sperm cells can become sterile. They will denature. The enzymes in the mitochondria will denature. The sperms will become inactive, or there'll be a low sperm count, or the sperms can be abnormal, or they will not be healthy. Therefore, the testes is protected by the scrotum because it's lying outside the body, or you can say the scrotum holds the testes outside the body to keep it at a lower temperature, or lower temperature, which means 2 to 3 degrees lower than the body temperature, for optimum sperm production, or you can say for healthy sperm production, or to say to ensure that all the male sperm cells are fertile and not abnormal. Okay. So now, again, it's about the male, and this one was really fully answered, and it was an all or nothing answer. What do I mean by that? You had to have everything right to get 2 marks. In other words, in life science, we do not give part marks. You either get the whole thing right, or if it's partly right, the whole thing is going to be wrong. So you have to be very, very careful of the way you answer the question. So this is a flow diagram. And with a flow diagram, you do not need a complete sentence. You use words and arrows to show a pathway. So a flow diagram is actually showing you the channeling or the pathway of something. So they're saying, show a flow diagram showing the pathway of sperm cells during ejaculation. Right now, learners? So now you had to talk about, if we must just go back to, let's see here. Nah? How can sperms move? Sperms can't move. If a male is going to release the sperm cell, it's not going to come directly from the testes. Because remember, what is this part here? That part above the testes is a coil structure. And what is it called? An epididymis. Epididymis. Nah? The spelling is very important. Epididymis. And don't confuse it with epidermis. If you're going to write something that sounds like epidermis, and although you're thinking it's epididymis, what you write on your script is you're going to get it wrong if it's spelled wrong. And this was what happened last year. It has to show epididy, the epididymis, okay? Epididymis. That's the coil structure. And what does it do? It stores sperms temporarily. It keeps extra, because every single day a male is actually manufacturing sperm cells by meiosis. So if he manufactures it every day and the sperm cells are not being released, it has to be stored. So it's first stored in the epididymis. It's not stored in the testes. So when the sperm cells leave the testes, it's going to go like this. It's going to go how? It's going to go from the epididymis from the epididymis to the vas deferens, which is the sperm duct, through the urethra in the penis, and then it's going to come out. Can you see that? So that's the pathway. The epididymis to the vas deferens, to sperm duct, to the urethra. And that's how it's going to be released. Okay? So this one was all or nothing. And they say, if the word testes is mentioned, no marks was awarded. So even if you had testes, epididymis, vas deferens, urethra, although it may sound a perfect answer, it's wrong, because when you ejaculate, the sperms do not come directly from the testes, but leave the storage place first. Okay, so it leaves the storage place, which is the epididymis. And now we're going to do investigative questions, which is a very, very big challenge to many learners, both in paper 1 and in paper 2. But there's certain things that are always questioned in investigations, and if you can master those criteria or questions and how to answer it, you'll do well. So whenever you have an investigative question, they always give you a table of data. You must analyze the table of data and relate it to exactly what is being said about the data. So it may look difficult, but if you read once and twice and relate it to the data, you will get the answer. So let's look at this now. Here is, before you look at the question, you're saying, here's a table. And then it's various plant hormones mixed in water. Now already you know, auxins, abscisic acid, gibberellins, that's my hormones. And they're mixed in water. So let's see, there's something with just water. Then there's abscisic acid and water. Then there's auxins and water. And gibberellins and water. So which 3 plant hormones are here? Abscisic acid, auxins, and gibberellins. Now in your mind, you must know, this one here, if it's only water and no auxins, it looks like that is my control. That if a change occurs, then it has to be because of the auxins. Because here, If there's no change, it's because there's no plant hormones at all. So that's my control, and these are my experiments. And then you sing height of the seedlings. That where there's water, it was 21 centimeters. Where it was abscisic acid and water, it seems like the height decreased. Where it was oxons and water, the height of the seedlings increased. But when it was gibberellins and water, it was 48 centimeters in height. Now remember that gibberellins cause the internode, the region between the two swellings, to increase in length and making the stem longer. And abscisic acid, remember, never ever gives rise to a length in stem. Abscisic acid brings about dormancy in seeds. They bring about leaf fall, the fruit to fall. Okay, so now we analyze this data and we look at the question. A student studied the effects of various plant hormones on the growth of corn seedlings. Now we know that the seedlings they're talking about here is corn seedlings. So corn seedlings, read nicely in the passage. Corn seedlings of the same height, and also use this word same with regards to species. Corn seedling of the same height and the same species were used. Now when something is same, it becomes your controlled variable. What does it become? It becomes your controlled variables. Whenever factors are kept the same, it is controlled variables, okay? And then you're going to have same height, same species, okay? And what you know that what does it contribute to? What do control variables contribute to? They contribute to validity, to keeping something fair. For an experiment, you have to keep something fair to make a comparison. You have to be fair. Then, so he had corn seedlings, same height, same species. Then he took 4 groups of 10 plants each. So basically, he took your corn seedlings and he divided it into 4 groups. Always picture what the scientist is doing. He took corn seedlings of the same height, same species, and he put them into 4 groups. And each group had 10, which means there's 40 corn seedlings. Right, now that you understand that one, all the initial heights of the seedlings were 1 centimeter. So when he started off the investigation, all of them had the same height. Is that important? Yes. So that should there be a difference in height, it must come from the experiment. Not you having one is 1 centimeter, the other one is 2. Now you don't know if the oxons caused the difference in height or was the plant already tall. So therefore, at the beginning, all the plants must be the same so that you can make a conclusion whether it was the factor that you're testing that caused the height of the plant to increase or was the plant already tall. Okay. And then you're going to look at the next one. Each group received the same amount of water. So what is the same now? Same height, same species of corn seedlings, same amount of water was given to them, okay? And all of them, when the experiment, were all of the same height. So same height even at the beginning of the experiment, right? And then she gave one group water only, okay? While the other groups each received 1 microgram of a different hormone mixed with water. So even the hormones, each hormone was of the same amount, 1 microgram. But only thing, you find that one of them had only water. Now, okay, so let's go to the next one. After 15 days, she measured the height of each seedling from each group, and then she calculated the average and recorded the information in the table below, and that is the table. So identify 2 ways in which the student ensured that the validity of the investigation. Remember, for validity we said same height, right? Or you could have said all the seedlings initially had the same height, or you can say that she ensured that all the seedlings used in the investigation were of the same height, the same thing. Same species were used, Same number of seedlings in each sample. That means there was 10, 10, 10. So the number of seedlings in each sample was the same. Same amount of water. Same height of the seedlings initially, and she ensured that all the seedlings were the same height. Same species. They belong to, all of them belong to the same species. And same amount of water. And same amount of hormones was given. And there was 1 microgram of, although the hormones differed, but the amount was the same. So same amount of hormone mixture. And she actually, after 15 days, she started to measure. She measured one after 5 days, another group after 10 days, another group after 12 days. No, she measured all the seedlings after 15 days. So she actually did the measurement at the same time. Okay, now we go to the eye. All right, and here we're going to look at the eye. And last year there were not many questions on the eye, so hopefully this year there will be lots more questions on the eye. And remember, the eye, the ear, the brain, and spinal cord has 54 marks. For you to pass a paper, you need 45. But eye, ear, brain, spinal cord, if it is mastered by you, you can actually pass life science just with understanding the nervous system. Right. So here's the diagram of the eye. What did I say? For you to answer a question, always analyze the diagram or the data table. Always know here, G is what? G is my yellow spot. What do I know? It has lots of cones for color vision, for clear vision. So I got cones and color and clear vision. That's my yellow spot. Okay. What is this one here? It's my blind spot, where there's no image, no rods, no cones. What is H? It's my optic nerve. Okay. It's my optic nerve that takes impulses from the eye to the cerebrum for sight to be interpreted. And then what is A? It's my ciliary muscle. What is B? It's my transparent lens. What is C? It is my aqueous humor. But if they were pointing right, it depends. You can say aqueous humor because it's pointing to the liquid. But if they drew a bracket in that space, if they drew a bracket and it's pointing to that space between those two, then that will be the pupil. But because they're pointing there, it's the aqueous humor. What is this D? This D actually is in line with the tough white protective coat called the sclera. So here's my sclera, which is opaque, it's white, and it extends in the front to D, which is my transparent lens. And then what is E? E is my suspensory ligaments. And then what is F? This is my fluid called the vitreous humor, which is jelly-like. Now I've analyzed my diagram on the eye. Now I look at the questions. Explain one feature shared by parts B and D that makes them suited to the function of clear vision. So now you must know, what is B? Oh, B is my lens. What is D? D is my cornea. Now, what is a feature or characteristic that is similar of the cornea and the lens? Oh, yes, both are convex. And when everything is convex, remember, explain, statement, and reason, or cause and effect. If the lens is convex, remember the lens is convex like this. Curved on this side, curved on that side. So if my lens is convex, even my cornea is convex. So both are convex. And what does convexity do in the eye? It bends light. Remember, light always travels in straight lines. So the minute it hits a curved surface, a convex surface, the light will bend to form an image on the inside of the eye. So both the lens and the cornea is convex for refraction. What else can be another feature? That is the cornea, and my cornea is transparent like a window pane for light to pass through it. Although it's coming from the tough white sclera, my sclera does not extend in front to be white. it extends in front to be transparent. So my cornea is transparent and even my lens is transparent. So I said one feature, I got two. I said both are convex and both are, because it's convex, then reflect light. Or you could have said that both are transparent to allow light to pass through. So any one of those two. If you just said both are transparent, you would have got 1 mark. Or both are convex, you would have got 1 mark. Okay. It's transparent, allows light to enter the eye and reach the retina. It causes refraction, bends towards light or bends towards the yellow spot. So it's convex and therefore it causes refraction. You have to say convex and therefore it causes refraction. See, both are convex to allow for refraction. Discuss how parts G and H work together for vision to occur. Now you remember you already labeled the diagram. So what is G again? G is my yellow spot. And what is H? H is my optic nerve. Now how do they work together to bring about vision or for you to see? So you're going to say that light stimulus falls on the yellow spot, which is rich with cones, and therefore it will be, the cones will be stimulated to create a nerve impulse, which will be carried by the optic nerve H to the cerebrum to interpret vision or sight. So here, children, you cannot talk generally and say the yellow spot has cones and which is stimulated by light to form a nerve impulse and carried by the optic nerve to the cerebrum. If you wrote all that but you didn't say what is H and what is G, it was going to be wrong. And that's why learners lost marks, not because they didn't know, but because they assumed I don't need to put down the name and the letter. Always put down the name of the letter and what does the letter represent in your paper, because that marker wants to know that do you know your work. And we don't know whether they want it in the memo or not, but you must ensure you're not there to defend yourself. So you must ensure you put everything down. And if the answer is perfect, they will sift out what they want. But if the answer is imperfect, you're going to get all of it wrong, or you may just get 1 mark. Right, so let's see. Discuss how parts G and H work together. What must you say? You have to say, you see that the light stimulates the photoreceptors in part G. Can you see? Which is the yellow spot. So the light stimulates the cones in G, the yellow spot. And then what's going to happen? It will convert that light stimulus to a impulse, nerve impulse, okay, to be carried by the optic nerve to the cerebrum to be interpreted, and that's how vision occurs. Okay. And then, so now you can see, learners, that investigations are not difficult at all. Okay, you will just analyze it. Everything is there, you just have to analyze it. So this one was a bit complicating because it had a lot of data available, graph, and you had to read, okay, so and you had to apply your knowledge. So now, an investigation was conducted to observe the time taken for the blood alcohol levels to return to normal. The time taken for blood alcohol levels to return to normal. Can you control someone's blood alcohol return, the time taken for the blood level to go back to normal? You can't. So automatically, you know that that must become my dependent variable. You look at when they say an investigation was conducted to determine or to observe, and then the time taken for the blood alcohol levels to return to normal. That is what you can't control. You don't know how long it's going to take. So what you don't know and needs to be measured is going to be a dependent variable on your y-axis. After the consumption of various volumes of alcohol, it seems like the various volumes of alcohol is I can control how much of alcohol I'm giving to the person and how long it's going to take for the alcohol to come back to normal. That's going to be my dependent variable. But the amount of alcohol that I want to give, maybe I want to give 10, 20, 30, what I can control becomes my independent variable. So it seems like the volumes of alcohol is my independent variable. Now let's see. Now they give you more details. Each participant was given a specific volume of alcohol to drink. Now, how many participants there were? Only 4. Now, children, in an investigation, you can't get less than 5. Less than 5 means a very small sample. And is that reliable? No. So the fact that this investigation only had 4 participants, it means it was not a reliable investigation. Why? The sample size was too small. So here's your 4 participants, and each one, the first one was given 30 ml of alcohol, second one 60 ml of alcohol, third one 120, and the fourth one was given 180 ml of alcohol. Now they tell you the blood alcohol concentration was measured before the participants drank the alcohol, and every 30 minutes thereafter until the alcohol concentration returned to 0. So every half an hour they were testing. I'm giving you alcohol, let's test. When will it go back to 0? Right now, they even gave a graph. So they gave you reading, they gave you a table, and they actually gave a graph. And they put here, how long did it take for the alcohol levels to go back to normal in the blood? So alcohol concentration, you can see here, when it was 30 ml, it took about 1 hour, maybe 15 minutes to go back to normal. When it was 60 ml of alcohol, it took maybe 2 hours and 15 minutes to go back to normal. When it was 120 ml, it took maybe 4 hours 20 minutes to go back to normal. But when they gave it 180 ml of alcohol, it took almost 6.5 hours to go back to normal. So the more alcohol you drink, the longer it takes for the blood concentration of alcohol to go to 0. Can you see it's taking, because there's more alcohol in the blood. So it's going to take even longer. So if somebody goes after Christmas and they drank maybe 3 glasses of alcohol, and somebody else maybe drank maybe a small little sip of alcohol, then the one who drank a small sip, by the time they go and drive, there'll be no alcohol in their breath or in their blood. But that one who drank 3 glasses of alcohol and then he goes and drives, if the traffic cops stop him and say, let me test the amount of alcohol from your breath, and they ask you to blow into like a spirometer to test the volume of the alcohol, it's going to take very long for him to. So if he just drank and he went after 1 hour, they would still get that the alcohol is still in the blood, so it's not safe for him to drive, so he's going to be fined because it never yet reached 0%. You see, like 0, it's still very long. Only if he drank it maybe at 12 o'clock, and if he goes home at 7 o'clock, then it will be fine. But if he drank it at 12 o'clock and he's going to go home at 1 o'clock, the alcohol concentration didn't go back to 0, so it's going to be in his blood and they're going to lock him up. Okay, so that is what this whole scenario is all about. Now you're going to look at this little perforation here. This area is showing you what range is acceptable for you to drink and drive, basically. What range of alcohol concentration is acceptable for you to drink and drive? After how long is it acceptable? Right. So now the question is, how many hours does it take for a person to be legally able to drive after consuming 180 ml of alcohol? Okay, let's just go back. Go back. Okay, children, remember when it was 180 ml of alcohol? I'm just trying to go back there. A minute. So you'll see it took 2.2. So here's your alcohol limit when it was like that. But now when it was that range there, when it was 180, for it to come to this range, this normal tolerant level, this tolerant range where they can tolerate your alcohol percentage, it was going from there and then it just came down there and it was 2.2 hours. Right. For this investigation, identify the dependent variable, and what did we say? It is the time it took for the blood alcohol levels to return to normal. Suggest 2 reasons why the investigator would ask the participant not to consume alcohol for 48 hours before the investigation. So if you're going to start off the investigation, we want the blood to be at 0% alcohol. We want the alcohol to only be in the blood from what is given during the investigation. Okay, so we don't want alcohol to be in the blood before the investigation because it's going to give a wrong value of whether there's a tolerance range or not in the person's blood. Is he able to legally drive now? Okay. So the 2 reasons why the investigator would ask the participants not to consume alcohol for 48 hours is that he must ensure that all the participants started the investigation with no alcohol in the blood or at a 0 level. To ensure also that it's fair, that you can't have alcohol in your blood and then also come and start the investigation. So to make the experiment fair and not to get false results. So to ensure validity, we say you drink alcohol only during the investigation. Do not drink alcohol and come for the investigation and have it in your blood when you come. Right. To ensure that all the participants begin the investigation with a 0 blood concentration. Okay. To allow enough time for the blood concentration to reach 0. So why is it, what's the question again? Why will you ask the participants not to consume alcohol for 48 hours? Because when you're doing the investigations regarding alcohol, we don't want someone to have alcohol already in the blood. So all participants must start off at 0 level to ensure validity. That's a plain, simple answer. That all should start at 0 to ensure validity. Now, this question was poorly answered as well. It's regarding the Airpods in the ear. Okay. And you can see, Airpods are wireless listening devices that fit into the ear and direct sound, allowing users to listen to music from their phones privately without disturbing others. So you find now there's a new trend whereby all the teenagers and lots of children, they have little like radio on their cell phones or music on their cell phones, and they don't want someone else to hear, and then they put the AirPods into the ears, and if somebody speaks to them, they don't respond, they can't hear what you're speaking. You've got to do that and get their attention, and then they will take it off and listen to you and answer you. Okay, so those AirPods are actually blocking the sound waves from reaching the eardrum. So now, a smartphone, if the AirPods is linked to a smartphone, it has a feature that can monitor the volume of music playing through the AirPods and gives a warning notification if the volume exceeds 80 decibels. Because if the sound is going above 80 decibels, now how you measure sound in decibels? So your eardrum or your tympanic membrane can actually withstand 80 decibels of sound. If it's going to be 120, your eardrum or tympanic membrane will vibrate so much that it can actually rupture. And if it ruptures or tears, remember it won't be able to vibrate, and little or no sound vibrations will reach the oval window, and then little or no sound vibrations will reach the fluid in the cochlea to cause pressure waves. No pressure waves, No nerve impulse is formed, and therefore no impulse will be taken to the cerebrum, leading to deafness. So people who listen to very loud music with the AirPods can actually become deaf if the music is too loud. But now the smartphone can monitor the volume. That if it's 80, it's fine, it's okay. But if it's 120 or 90, it's not okay, because now you are reaching a stage whereby your tympanic membrane can burst. Right. Explain 2 ways in which AirPods could prevent a person from hearing other environmental noise. So let's say, for example, a mother is busy and she's listening to music and she got the AirPods in her ears, and then she can't hear that the baby is screaming and crying. She's busy dancing and washing her dishes. She will not be able to hear the baby crying, or any other, or her husband came into the yard, or there's someone that came. She won't be able to hear if a dog is barking, because the sound from the AirPod is directly next to the eardrum. So obviously the sound from the AirPod is going to be louder than the environmental sound. So the sound waves from the AirPod will be louder compared to the sound waves coming from the outside. 2, the AirPods are like an obstruction. It is preventing the sound waves from the outside from reaching the eardrum. And that's why she won't be able to hear. Right, so let's see. So, how are the 2 ways in which AirPods can prevent a person from hearing other noises? The AirPods block the auditory canal. They block it. So remember, now you have your pinna, and here's your sound waves coming from the environment. Here's your auditory canal, and here's your eardrum. Now you put in an AirPod here. That's your AirPod. Okay? And then it's going to go there, and there the AirPod is blocking the sound. So the sound waves from the outside is being blocked in the auditory canal, and it's prevented from reaching the eardrum. That's one way. And the other way is the sound from the AirPod. Now this sound here is more loud. The sound of the AirPod is too loud, and the environmental noise is too quiet, so it's not detected. So the sound from the AirPod is going to be louder than the sound from the outside, because it's closer to your tympanic membrane, and that's why the person will not be able to hear. Therefore, what is advisable is that people cannot put 2 AirPods in both the ears at the same time. All environmental noise will be blocked out. It's actually dangerous for them, because even somebody is screaming and shouting for them, they will not be able to hear. So it's better to put 1 AirPod in one ear and at least leave the other ear for environmental noise or for other dangers, for them to be alerted. Okay, so this is how this question is actually interpreted. So it may look difficult, but actually if you think about it, it's about the eardrum, it's about the tympanic membrane, it's about the sound waves, right, and reaching the eardrum. Right. So each point must have linked a cause and effect. For example, the AirPods block the auditory canal. You can't just say that. The question is, why will the AirPod prevent a person from hearing other environmental noise? And you say AirPods block the environmental noise. We'll say it blocks the auditory canal and therefore prevents the sound waves from the environment from reaching the. It's actually like a blockage in the auditory canal, so it prevents the sound waves from reaching the eardrum. Okay, and then also that the environmental noise is too soft and you can't stop there, because the sound from the AirPod is even louder than the environmental noise. So that's how you got to give a cause and effect. So this is a question, learners, from the Free State Prelim Paper 1 of this year, right? A defender in a netball game prevented a goal from being scored when she jumped high to catch the ball. So now we don't know what topic is this. A defender in a netball game prevented a goal from being scored when she jumped high to catch the ball. As she jumped, there was a change in the direction and speed of her head. Immediately, what do we know? This got to do with the ear and it got to do with balance. Okay? How do we know that? Change in the speed and direction of the head. Not just speed and direction of the head. Change in the speed and direction. Because she was like this and then she jumped in the air to catch the ball and obviously she must have fell. So there was a change in which the head was positioned and directed, okay? And the speed as well. Describe how she maintained balance as she jumped to catch the ball. So how did she maintain it? Because she's jumping to catch the ball and she can fall. So how did she catch the ball and also maintain her balance? Right, so let us look at that answer there. First of all, you got to say, learners, remember that with balance, you got to think of the cerebellum, you think of the skeletal muscles, you think of speed, because remember our ear is in our head. So how we move our head, how fast we move our head, the speed, and which direction we move. You're looking up at the ball and you're catching it and then you're going down. So it's how fast, speed, and the direction of the head, because your ear is in the head. So change in the speed and direction of the head causes movement in the fluid. Where the fluid? In the semicircular canals. Remember, we have your semicircular canals like that, okay? Now here is your ampulla and here's your semicircular canals. And what are they filled with? Fluid. So now what is going to be here? It's going to be your cristae. And what nerve leaves? It's going to be your auditory nerve. And where does it go to? The cerebellum. So now you want to know, describe it. How did she maintain the balance? You've got to say, a change in the speed and direction of the head will cause the movement of the fluid, which stimulates the cristae. The cristae is what? A receptor. In the ampulla, or you can say in the semicircular canal, the pressure wave was converted into a nerve impulse. Remember, inside the inner ear, there's pressure waves. Out ear, sound waves. Middle ear, sound vibrations. Inner ear, pressure waves. So, when you change the speed and direction of your head, the fluid in the semicircular canal will stimulate the cristae, and then it's going to stimulate and cause a pressure wave. And then that pressure wave will be converted into an impulse, which is a message to be carried by an auditory nerve to the cerebrum, also to the cerebellum, where balance is interpreted. Why cerebellum and not cerebrum? Remember, cerebrum got to do with your interpreting your senses, got to do with your voluntary action, and it got to do with interpreting what? Voluntary action, it got to do with your higher order thought processes and interpreting your senses. But cerebellum got to do with balance, posture, and muscle tone, and voluntary movement, right? How you are moving, how you are jumping, and how you are hopping to actually catch the ball. So cerebellum got to do with balance. So children, please understand that whenever they talk about maintaining your balance, you must talk about a change in the speed and direction of the head. So even if you're going to trip over a stone, change in the speed and direction of the head will cause the movement of the fluid. The fluid where? Inside the semicircular canal, which will stimulate the cristae. This will cause a pressure wave. The pressure wave will be converted to a nerve impulse. It will be transported along the auditory nerve to the cerebellum. And then the cerebellum now in the brain must send a nerve impulse wave to the muscles of your bones, which is your skeletal muscles. And your skeletal muscles will contract and relax, and you're able to move your head and balance your legs to catch the ball without you getting damaged without you actually hurting yourself. You'll try to somehow roll and or do something to maintain your balance. So balance is simple. What are the key words? Change in speed and direction of the head causes the movement of the fluid in the semicircular canal, stimulates the Christie, which sends a nerve impulse, right, to the cerebellum, which sends a nerve impulse to the skeletal muscles. And then balance is going to be restored, to restore your balance. Okay. Now we're going to have another question here, and this got to do with the kidney. Okay, here's your kidney, and here is a gland. What is the name of the gland on the kidney? It is called the adrenal gland. And here's your kidney. Learners, immediately remember, what are the two hormones that the adrenal gland secretes? It's adrenaline. And what is adrenaline used for? For emergency. And the other hormone that the adrenal gland secretes is aldosterone. Already we analyzed. Aldosterone is released by the adrenal gland, and it goes and stimulates the kidney tubules to maintain your salt balance. And adrenaline is released in an emergency situation, and it causes my heart to beat faster, my pupils to dilate, my bronchioles to dilate, my breathing to increase, my glycogen becomes converted to glucose, my glucose levels will increase when adrenaline is released, because I need glucose, I need energy to run away from something that's frightening me. I've analyzed this. I know that this question got to do with endocrine and homeostasis. I know that X is my ureter. It is a tube that transfers urine from the kidney to the bladder. And I know that my kidney tubules controls my water balance. And the water balance is controlled by the hormone ADH. So now I've analyzed the whole diagram. And then I look at the question. The diagram below shows a kidney, its blood supply, and the ureter. Explain what the homeostatic response. Remember, children, these are the difficult questions found in paper 1. Okay, so when it's difficult, you have more explaining questions, more data interpretation, okay, more analyzing of graphs. So for this, explain what the homeostatic response was when urine is diluted in X. What the response was to cause the urine in X to be diluted. First of all, do you understand what is dilute? Dilute is the opposite of concentrated. So if I say I want my juice to be diluted, it means if I buy Oros, which is a concentrate, and I put, for example, 2 tablespoons of Oros juice into a glass, and I fill it with water, only 2 tablespoons. That means I have more water than juice. It means it is diluted. It's very weak. But if I have a sweet tooth and I say I'm going to put half a glass of Oros into a glass, I'm going to put half of the glass must be the Oros concentration and just a little bit of water, then that's going to be a very, very concentrated juice. So when our urine is concentrated, it is very dark yellow and it has less water. When your urine is diluted, it means it's very pale yellow. It's very, very, very pale, almost colorless, and it's diluted. So the more water the urine has, the more diluted it is. The less water it has, the more concentrated it is. Now they want to know, in X, the urine was diluted, which means this person is excreting urine with more water. Now what could be the reason? It's because his water level in the blood was already high. So he can afford to lose water in his urine. So let us look at the answer. If the urine is diluted, you must tell yourself, oh, this person is excreting more water in the urine, which means that his water level in the blood was high. So let's see. Water level in the blood is above normal. Or you could say it was high. Or you could say the water level in the person's blood was increased. High or above normal. That's the first thing. Then what happens? Because his water level was high in the blood, the receptors, you must remember, something must receive that message. The receptors in the hypothalamus. The hypothalamus is in the brain. So the receptors in the hypothalamus are stimulated. So because the water level is high in the blood, a nerve impulse goes to the hypothalamus. And the hypothalamus is able to detect, oh, this person drank a lot of water. He drank juice, he drank water. His water level is quite high in the blood. We do not need to reabsorb more water in his blood. So when the water levels are high in the blood or above normal limits, the receptors in the hypothalamus will be stimulated, and it's going to send a nerve impulse to the pituitary gland. So remember, that is the brain, and here is your corpus callosum, and there's your hypothalamus, and there's your pituitary gland in the brain. Okay, so remember that. That's how the brain is. Okay, so here's your corpus callosum, and here's your hypothalamus, and here's your pituitary gland. So the water level is high in the blood. It will stimulate the receptors in the hypothalamus, which will stimulate the pituitary gland to secrete what? To secrete, when we talk of water, the hormone ADH. Ne? So it will send impulses to the pituitary and tell the pituitary, do not release ADH. Because what does ADH do? ADH makes your kidney tubules permeable to water, allows water to leave it, to go back into the blood, to make it permeable. It's normally impermeable. But when ADH is released, the tubules become permeable. Now you have enough water in your blood. So there's no need for water to leave the kidney tubules and go back into the blood. So because there's lots of water in the blood, the high levels of water in the blood stimulates the pituitary to secrete what? Less or no ADH. To stop secreting ADH, to secrete no or less ADH. No or less ADH goes to the blood in the kidneys, right? Because remember, ADH is a hormone and it's traveling by the blood. The renal tubules or the distal tubules or the kidney tubules become less permeable to water. So you find that the renal tubules become less permeable to water because there's no ADH, and therefore less water is reabsorbed into the blood, and the water levels in the blood will decrease and go back to normal. Because it was already high, so by not reabsorbing more water, the water level will come down because more water was excreted in the urine. Okay. Now we want to know of a drawing. Learners, they can ask you, there's definitely going to be a drawing in either paper 1 or a drawing in paper 2. So here they say, remember, a drawing is a skill. So you must have a sharp pencil. Your labels must be in pencil. The label lines must be in pencil, and your labels itself must be inked. And you must have a heading or a caption for your diagram. So make a label drawing of the parts of the eye that are responsible for accommodation. And in your mind, what must you know? Accommodation is the ability to focus on far and near objects. What are the parts? Oh, it's my ciliary muscle, which has my suspensory ligaments that attach to my lens, and I got my ligaments and my ciliary body, something like that. So those are the parts that help me to see far and near. Remember, what happens during near vision? Remember, when you're writing your paper is near you. So C, ciliary muscles will contract. Do not get confused between circular muscles in the iris that got to do with controlling the amount of light that enters. Iris is circular, like a bicycle wheel, and radial. But distance, will focus far near, is ciliary muscles. Right, so the ciliary muscles will contract. These ligaments are going to relax. What are the ligaments called? Suspensory ligaments, and not just ligaments. Because you get ligaments between the bones. So we're not talking about those ligaments. We're talking about the suspensory ligaments which are holding the lens to the ciliary muscle. And there is my glassy lens, my transparent, elastic and biconvex lens. And that's going to help me to focus far and near. Now, learners, before we look at the diagram, let me explain also what are the 3 characteristics of the lens that helps you to see far and near. First of all, remember, they can ask you this question end of the year. That the lens is elastic. Okay, it's elastic. So it can become more convex and less convex. More, not just convex, it's already convex. It's already convex like that. So it can become more convex or less convex. More convex or less convex. So more convex for near, your suspensory ligaments will be loose. Ne? Your ciliary muscles will contract. So remember, sometimes they can ask the question, make a label drawing of parts of the eye that are responsible for accommodation when a person is looking at a book close to him. Then you're going to draw a ciliary body with suspensory ligaments that are loose, with a lens that's very, very convex, with suspensory ligaments that are loose, and your ciliary muscle here. And you label here, ciliary muscle contracts, suspensory ligaments loosen, lens more convex, near vision. You see? But now they're just asking you for make a label drawing of the parts of the eye responsible for accommodation. So you don't have to label what is the changes in that's happening to the parts. You just have to now label the parts, and that's how it's going to look. That's my ciliary muscle, that's my lens, and that's my suspensory ligaments. Did I answer the question? Make a label drawing of parts of the eye responsible for accommodation. They didn't tell me near, they didn't tell me far, so I must just label the parts that are involved. A ciliary muscle, a suspensory ligament, and a lens, and then obviously I must be able to write down the heading, parts of the eye responsible for accommodation. Every diagram must have a heading, and diagram like this, say it's a correct diagram, you get 1. Any 2 labels, you get another 2. Ne? 1 mark for caption, you get another one, 4. Alright, so you can get 4 marks for a simple little diagram. Alright. And then we go to another question here. The diagram below shows parts of the male reproductive system. Again, same diagram. It looks a little different, but the parts will always be the same. Again, there's the penis, there's the urethra. This diagram is even better. It shows you the testes, it shows you the epididymis, it shows you the scrotal sac, it shows you from the epididymis comes the vas deferens. See, the sperm cells that were stored there goes from the vas deferens, then it collects the fluid from the seminal vesicle, from the prostate gland, from the Corpus gland. It comes through the urethra, and then remember also sperm cells are going like that. The sperm cells will now float in the fluid of the 3 glands to call it semen. Fluid plus sperm cell is semen. It will go through the urethra and out. That's how it's going to be ejaculated. So now, prostate cancer affects men, and the risk increases as men get older. So many women have breast cancer, ovarian cancer, but in male, the cancer that is very dominant is actually prostate cancer, the gland below the bladder. And the only way to get rid of it is to actually remove the prostate gland. Removal of part B. You remove this prostate gland, which is shaded in black. Explain how removal of part B may affect male fertility. So if you remove the prostate gland, ask yourself, what is the function of the prostate? The prostate gland secretes an alkali fluid to neutralize the acidity in the vagina. Therefore, if it is removed, the acidity will not be neutralized, and this will denature or kill the sperm cells and lead to male infertility. You see, it's going to lead to what? Not male fertility, male infertility. So let's see again. You say, explain how part B may affect male fertility. Tell yourself, part B is a prostate gland. It secretes an alkali fluid to neutralize acidity in the urethra or vagina. Therefore, if there's no prostate gland, there'll be no alkali fluid to neutralize the acidity in the urethra, and this will kill the sperm cells or lower the sperm count, leading to male infertility. The male will not be fertile. Sperms won't be fertile. See, alkali fluid will not be secreted to neutralize the acidity in the urethra or vagina. Sperm cells will denature or die or become abnormal, and this will decrease male fertility or lead to male infertility. Okay? And then here's another question about a sperm cell. You can see a sperm cell here that the head is torpedo shaped. You must draw the acrosome, which is C. That's the nucleus, which contains the genetic material. That's the neck with the mitochondria and a very long tail. All right. Explain 2 ways in which the sperm is structurally suited to ensure effective movement towards the fallopian tube. So how is this adapted to move towards the fallopian tube in the female? 1, it contains a mitochondria for what? Cause and effect. Cause and effect. It has a mitochondria. You can't just stop there, because it's got to do with explaining that this sperm cell has a mitochondria that supplies energy for locomotion of the sperm. What's locomotion? Movement. Because they are saying, what are the ways in which the sperm cell can move towards the fallopian tube for movement? So it contains mitochondria to give it energy to move. It has a long tail to enable movement. It has a torpedo-shaped head to allow movement without causing friction. Can you see that? It's torpedo-shaped to prevent friction, so it can move without causing friction. Explain the consequence for reproduction if a sperm cell does not have part C. So I'll tell you that. C is the acrosome. It secretes enzymes to digest the membrane of the ovum. Therefore, if there is no C, there is no enzyme to digest the ovum membrane, and therefore the nucleus of the sperm will not be able to fertilize the nucleus of the ovum. There will be no fertilization. And then that's how you explain that no enzyme will be present. The sperm will be unable to penetrate the ovum or the outer membrane, and therefore there's going to be no fertilization. Explain how the male body ensures that the sperm cells are not killed by acidic urine as they travel through the urethra. How does the male body ensure that when the sperms go through the urethra, which also passes out urine, the urine can be acidic? How does it ensure that acid of the urine will not kill the sperm cells? Remember the prostate gland. The prostate gland secretes an alkali fluid to neutralize the acidic conditions of the urethra. And then here's the brain. Explain why damage to part E can lead to immediate death. And you can see, children, E. Now that's the pons, that's the spinal cord. And between the pons and the spinal cord is what? Our medulla oblongata. So explain then you must say, medulla oblongata is responsible for vital processes such as breathing and heartbeat. Né? Therefore, if the medulla oblongata is going to be damaged, the person will not be able to breathe, to take in oxygen for respiration, and his heart will not be able to pump glucose and oxygen to the cells, and it can cause organ failure. You can see the medulla oblongata controls vital processes such as the heartbeat and breathing, and therefore these processes will stop, and then there'll be no glucose and no oxygen, no respiration, no energy, and that can cause organ failure leading to death. Okay, so make a labeled drawing of a neuron that carries the impulse from part A to the muscle. Remember, part A is part of the central nervous system. So which neuron takes the message from the brain or spinal cord to your muscle? M for muscle, M for motor neuron. And it's going to be that neuron. And you can see this neuron here. It has a star-shaped cell body, many extensions called dendrites. There's the cell body, there's the nucleus, there's the axon. You cover it with the myelin sheath. You can even do it like that. That there. Then here's your axon, then here's your myelin sheath, right? Here's your axon nerve endings, and here's your dendrites, and there's your nucleus, and you can draw your neurolemma, right? And always the impulse moves in that direction, which means from the dendrite, cell body, and axon. And there's your myelin sheath here, and that is your axon nerve endings, your dendrite, your nucleus. That whole thing is the cell body. This whole thing is the axon. At least 3 labels. And plus, diagram. What must you write here? Diagram of motor neuron. You must have a caption for your diagram to get a mark. This one was a bit difficult, learners. It was also from the Gauteng Prelim Paper 1. And here it's talking about basically how the scientists wanted to investigate the hours that people spend at the work, long hours where they are sweating. And if they're sweating, they're losing water. And if they're losing water, it can affect the mental and physical performance and pose a risk to their health. Right. So here we found that male subjects, they were exercising and they wanted to find out their temperature. So give the water loss range that the largest number of males fell into under summer conditions. Now, you know, if people are working outside for long hours and they're sweating, they sweat more in summer when it's hot. So what is the largest, what is the highest water loss range or the largest number of males that fell into? So you can see that the highest number of males, the largest number of males that fell into which water loss? So you go for the highest loss of males, that the highest number was actually 10, and then the highest water loss there for 10 males, that means was between 0.4 and 0.49. Okay, so in this case, you simply had to read. They asked you, give the water loss range and not the number of men. Give the water loss range that the largest number of males fell into summer. So summer is the key. It's the light shaded one. So you're going to look for the light shaded bars. That represents summer. You're going to look for the highest number of males. That one is 10. And what was the water loss range there? It was 0.4 to 0.49. So you can see, you look at the question and then you correlate it to the graph. Summer, I look at the unshaded one. Males, the highest, I look for the highest. And then look for the water loss, because they said give the water loss range. Okay, and that's how you analyze that graph there. Same question. Describe how the body of a healthy person will try to prevent dehydration when the water levels in the blood decreases below normal. It's simple. If the water level decreases below normal, means they are dehydrating, they are losing more water by sweating. What's going to happen there? You're going to simply say, when the water levels are below normal, the hypothalamus will be stimulated, This is going to be the opposite of the other one we did. It will send impulses to the pituitary to secrete more ADH. And then the ADH is going to make the kidney tubules more permeable to water. More water will be reabsorbed into the blood, and less water will be passed into the urine, and therefore the water levels will be returning back to normal. Right, see, less urine, because so when you're sweating a lot, you can't also urinate a lot. So if a person is sweating a lot in summer, how do you maintain your water levels? It means because you're sweating, the water levels are low, more ADH, pituitary secretes more ADH, more water reabsorbed from the kidney tubules because they become more permeable, more water reabsorbed into the blood, and less water will be excreted in the urine. That means that urine is going to become concentrated. Explain how the change in the average loss of water through sweating from winter to summer affects the body temperature. Now, children, look here. If you are going from a change from winter to summer, how is your body temperature going to be affected? Remember now, is becoming very hot. So when it becomes hot, sweating increases. The body temperature is going to decrease. So they want to know, how will the change in the water loss through sweating? Now, how does the change in the average water loss from sweating affect the body temperature? Because you're sweating in summer, what's going to happen? More sweat, more evaporation. Okay? More sweat, more evaporation, and the body temperature will decrease because there's more evaporation. Right. Then what are the 2 ways that you can do to ensure that your workers don't dehydrate? You can provide them with shifts in the morning for shorter hours. You can provide them to wear cooler clothing, and you can also ensure that you provide them with cooler water. Okay, children, and here is a simple diagram to show you the liver and the pancreas and how they control the glucose level. And you can see that the pancreas will secrete insulin, which will convert excess glucose into glycogen. See? Explain how the lack of hormone affects the blood glucose level. If there's not enough insulin, the pancreas will secrete little insulin, right? So if there's lack of insulin, lack of hormone C, little insulin, no insulin will go into the blood, and then little insulin will reach the liver, and therefore the excess glucose cannot be converted into glycogen. And then that is where we will stop now, right? And we've covered endocrine and homeostasis, nervous system, male reproductive system, reproductive strategies. So all the difficult questions were actually covered in this paper. So good luck, learners. The more you go over your questions and you go through the answers, the more you will be able to answer higher levels of difficulty questions. Thank you.